Welcome to the Silicon Slopes podcast. I'm here with Adam Brown, who is the CEO of Quansys Biosciences. How are you?
I'm doing well, thanks. Thanks for having me.
You bet. Thanks for driving down all the way from beautiful Logan.
That's right.
Appreciate it. All right, so we've got Quansys Biosciences, and this is going to be fun because it's something that we haven't talked about a lot. We've had a couple of companies in this field on the podcast, but not a lot. So if I screw up any of the acronyms or pronunciations, you'll let me know. But what is Quansys?
So Quansys Biosciences is a company that develops and manufactures what's called an ELISA. It's a test that measures and detects different proteins that the body produces. And we have taken that technology and moved it forward where we'll test multiple biomarkers that the body produces simultaneously at one time in an assay or test. Yeah.
The scientific term is assay.
That's right.
We'll just call it test.
Mm-hmm.
Don't have to beep anything out. Okay. So the application of your guys' products is?
So it's pretty widespread where it can be used, but a lot of researchers use it in, academics in different research areas. So if you're looking into cancer testing, the body has an immunological response to just about everything. And so, the biomarkers are produced if you have cancer, if you have a cold, anything like that, your body is having an immune response. And so our tests or assays can be used in research, they can be used in drug development, they can be used in quality control, all over surveillance to see how a population has responded to drug treatment, or, in case of COVID, it's used to kind of get a general idea of how many people may have been exposed to COVID in a population or different things like that. It's really broad in ways that it can be used.
Yeah. And so that data piles up pretty quickly, right?
It does.
And I assume that's been like the problem that a lot of folks have been trying to solve for the last couple hundred years is how to maximize that data and make decisions quicker and better.
Right. So, I mentioned the surveillance studies. We work with a nonprofit based out of Washington called PATH. And that's like their primary purpose. They go to third world countries. They look at diseases that are prevalent, malaria, micronutrition, and they'll want to study that population, collect that data, make sure that things are progressing in a way that is making real improvements. And yeah, so a lot of data backs that on that end, and getting those tests that are accurate is very important.
Yeah. And so a couple of the examples of end uses or end cases, what would be some of them that popped to the top of your head?
It's really on that research side is where we see a lot of it. We used to put together these little conferences where we would have some of our customers come in and talk about some of the things that they had used our tests for. And, so one of them was with a university, and he was studying mice. And if you punch a hole in the ear of a mouse, it will regenerate and heal itself. And humans don't do that. So, he was using our assays to test the response of a mouse versus a human and what kind of things would interfere with that regeneration. A lot of the biomarkers that we use are called cytokines, and they are inflammatory type responses that your body puts out, and those inflammatory responses can actually inhibit that type of thing. So he used that for that kind of research of, hey, regeneration in a mouse, where one of our biggest customers, we've developed a test for what's called the complement system in your body, and they go out and they sell that to a lot of drug researchers and people that are trying to put together therapeutics and things that can actually cause or provide good ways to just enhance general health. And there's so many uses, it's hard to say what some of those end uses are, but hopefully that helps a little bit.
Yeah. So it sounds like it's proactive in like research and trying to get ahead of problems, but then also retroactive if, there are sicknesses and you need to do the tests and do the data. So it goes both ways, it sounds like.
Yeah, it does. And the advantage, as I mentioned before, I brought this. This is how our assays are run. We have these little plates with each of these wells.
Yep.
This way.
Yep.
Historically, each of these wells would be an individual test or assay. And what we've done is we'll spot micro spots in the bottom, up to 18 right now. And each of those markers is its own test within the well. So you're getting more data out of one test. You can use less sample. Blood is used a lot. Saliva can be used, all different types of things from the body. And you can get a better answer quicker. So, if you were looking into diabetes, there might be 6-7 different diabetes markers that a researcher would be interested in. Instead of running 6 or 7 different tests, in a row, we can do it all at once. And that's a nice benefit for those people to get faster results and save money.
Yeah. So, you kind of see some footage on the nightly news of somebody developing a research or a pill, whatever it is. And there's always the person dropping a little something into one of those.
Mm-hmm.
And so you've basically times it by 18 with each little drop point, right?
Yeah, with each of those spots.
Yeah.
Mm-hmm.
And so that's obviously a really great product and a force multiplier in the research and development. What other products do you guys have, and what's kind of the software component and the data component to it?
Yeah, so this really is the main product, but there are different aspects to make it work. If you were to just squeeze stuff down on the bottom, it wouldn't get you anything other than you would have a wet plate with the holes in it. So the way that it works is there's actually what's called a chemiluminescent reaction. So each of those spots will actually start to glow when different components are put together. And we've developed what we call an imager. It's a box that we put this inside, and it images those spots over a period of time to collect that light, where it's connected to a software program that we've put together that will take each of those light sources from each of those spots. And then what it does is it'll analyze the intensity of that light and give it a numerical value. And then that software will analyze all that data. And it's a big mathematical spreadsheet that the researchers get at the end. For most people, it's just a bunch of numbers, but look at that. It'll show a little graph with a range and a curve to that. And that's what they use to actually give value to it. This by itself is nothing without that software program. And it's actually kind of the crown jewel of what we do. The software program is really user friendly. It's able to just generate a lot of data that researchers want to use.
Yeah. So those types of answers blow my mind, right? So all that work that went into that, you've got to have the scientists that understand diseases and human body and all of that. But the data starts with light. And
yeah.
You need the software and the computer engineers to translate that, somebody that's good at math. But what you just described is like an amazing process.
Yeah, it's pretty cool when you think about what goes through it. That first step of, well, what markers even need to be looked at, and the science that goes behind that, putting those together, building the assay. Our R&D team, they'll spend months sometimes making sure that this part of it even works, and then testing it out, making sure that we're getting enough light to actually capture that and get an accurate response, and then making sure that software program is pulling accurate data is critical.
Yeah. Yeah, there's not probably a lot of redos in your guys' industry. If you have a bad reputation that the data is always wrong, then you'll probably be blacklisted. Is that how it works?
Pressure's on. Yeah, for sure. A lot of researchers use sample from samples what we put in that actually has the information we're looking for from mice. And when you think about a mouse, they're really small.
Mm-hmm.
There's not a lot that you can pull out of that. And so if you mess that up, the chance of trying it again and again is really limited. So we need to make sure that the quality is there and the results are given correctly the first time.
Yeah, because your end users, your customers, they have their own budgets, they have their own time constraints, they have their own personalities, and efficiency is key for them, right?
It is.
You saw that with like how quick the vaccine came out, right? I imagine it gets pretty intense for a lot of your end customers to deliver their results to their company, their shareholders, their university, whoever it might be.
Yeah, definitely.
Very cool. Okay, so you guys are based out of Logan, right?
Correct.
And for the company and the team that you're building, what types of folks are you looking for to join the team? What backgrounds do they have? And then how do you cobble together a team that needs to sell the product and market the product, but you also have the scientific side of things? How do you manage that as a CEO?
Oh man, that's the challenge I think for any business owner or CEO, someone that's running the business, is how do you get the best talent to fit those roles? You know, for us being in Logan, we are connected well with Utah State University, and we work with them to get in biological engineers, different chemists that can come in and work on that science side. But then also, just like any other business, we need to find people that can sell and market that product. And for us, it's actually really difficult to find people that have that science background and the business background to come in and be able to say, oh, I understand what this product is, and I can help explain that and sell it to somebody else. And that's an area that we focus a lot on, make sure that we can get good reps that come in and can share that message on both marketing and sales, but then getting the team members that can come in and produce that product is important too. So that relationship with Utah State University has been really helpful. But yeah, just young people that are looking for, I don't know if young is okay to say, anyone, right? All ages. But yeah, anybody that can come in. But we work a lot with young people that are just starting their career and trying to get things moving and going. And we see a lot of those.
Yeah.
People.
So in addition to all your other responsibilities, do you find that you're often kind of in between the two, like the business side and the researcher and scientific side, and do you have to translate back and forth occasionally?
Yeah, we're a smaller company. We have about 30 full-time employees, so I find myself in different areas. I'm definitely not back moving liquid, and I don't understand all the ins and outs of that. My background was business, not science, actually. So I've had to learn a lot while I was there. But I'll go back and spend time with people in the lab and, cap some bottles here and there and stuff like that, just to get involved and make sure I have a good understanding of what's happening as well.
Yeah. So, unless you've been hiding under a rock, there's been a pandemic that a lot of people are interested in, even if they're just like a Monday morning quarterback. A lot of scientists out there these days. But what did that affect your business like?
So it actually made it easier for me to describe to people what we do, because with COVID, which I'm assuming is what you're talking about.
Yep.
But with COVID, we actually developed an antibody test for COVID-19. So it would detect the different, it's called the spike 1 and spike 2 proteins or antibodies against the spike 1 and spike 2 protein that your body produces if it's been infected or come in contact with COVID-19 or SARS-CoV-2, the virus itself. And so, we'll take a drop of your blood. We can test that, come back and tell you if you were exposed. And then we actually added to that by adding what was called the nucleocapsid. And with the nucleocapsid, the spike 1 and spike 2 protein, we can tell if you were infected naturally or if you have built up an antibody response based off of vaccine. So in a population study, we could go test a large population. We're working with the University of Minnesota right now and NORC. It's a Northern I can't remember the acronym, but they're based out of Chicago, a research group where we're doing that exact same thing in Minnesota area, testing populations, saying, okay, these people have been infected, these people have been vaccinated, gives us a general idea of what that looks like. So we've done some stuff there on the COVID-19 side, and that's been exciting for us to work with and on.
Yeah, I can imagine. And so from kind of start to finish, like once you guys kind of figured this was a real thing, and that you guys were in a unique position to help out, what was that process like?
Early on in probably December, January of 2020, we started hearing the news of things going on in China and this virus moving, and we had some initial conversations. At first, we weren't sure if what we had would be applicable early on, and so we had some discussions back and forth then, but quickly decided, hey, this is an area that we can make a difference if we do something. So we developed that assay in, I think, 6 weeks, which was really quick for us.
Yeah.
Luckily, it wasn't too complicated, which helped. But we had that, submitted it to FDA for emergency use authorization, and we received authorization. Unfortunately, that part was the longest part.
Shocking.
We got that in October.
Oh.
So it took months to get the authorization. And at that point, a lot of the focus has been on testing and current infection. So our test won't tell you if you're currently infected with COVID-19, but it will tell you if you were in the past.
Yeah.
So that was a little bit of a hit for us because the focus was definitely on that other end rather than this end. But now we see interest on the surveillance study of just populations and what's going on, and that's where we've seen the most interest with our test.
Very cool. Yeah, I've been on the timeline of your, the company's history. That'll be a kind of a proud point that you were able to circle the wagons and be additive to the whole thing.
Definitely.
Very cool. All right. So, just like every other CEO, you've got to look to the future as well. How do you strategically think through the future? Is it products? Is it just refining what you already have? How do you view that?
It's a little of both of those. So one of the things that we're working on right now is bringing in-house some of the process of developing our assays. So we use antibodies and antigens to develop these assays. And historically, we purchase those from third parties, bring them in, use that to develop. So we're actually in the process of putting together our own lab where we can what's called express antibodies within our own lab. So that'll expand our services. We can offer those to customers as well. It helps with the performance of our assays over time. Just make sure that's more consistent. So that's an area that we're looking into and working on. Another thing that we're doing is, I mentioned there's 18 spots that we can fit in the bottom of the well. We're onboarding some new technology that will allow us to raise the number of spots that we put in there. So printing wise, we'd get more spots in. Biology wise, it'll definitely be more difficult to get more in because they don't always like to play well together. But that's an area that we're working on as well.
Yeah. Yeah, that's kind of one of the components that it's intriguing with this company is there's certain laws of nature, right?
Yeah.
And they're pretty well established.
Sometimes those biological markers don't play well. So you could have two spots in there and they just will not work together. And that's where a lot of the challenges come in on that development side, is making sure that we can actually get them to perform the way they're supposed to.
Yeah. And so customers, what is a good sign of customer success for you guys, and how do you kind of track and monitor that?
A lot of our customers are OEM customers, and so they'll come to us with specific biomarkers that they want to look at or resell. And, we've seen a lot of success working with them and developing those assays or tests for them. And that's been a big focus for us. And so to make sure that they're successful and happy is making sure that the test performs as it's supposed to, that we're able to get as many of those markers in as possible. One of our really good partners that we've developed assays for, they came in with 9 markers. We developed that for them. They went out, they're reselling it with great success, came back, and we want to expand this. So we added, 6 or 7 more markers to that. And then, seeing that kind of thing where they come to us, they see something work, they do it again and again and again. And we're working with a lot of good partners in that way. And that's been a real key to our growth.
Very cool. Last question. It's kind of the meat and potatoes of it. You mentioned right before we pressed record that you have a restaurant.
I do, yeah.
What's it called?
It's called Stacked Quality Pancakes.
Stacked Quality Pancakes in Logan, Utah?
That's right. Downtown on Main Street in Logan.
Very cool. So what similarities are there with your role as CEO of Quansys versus restaurateur?
It's very different. I actually started with the restaurant, and I got involved with Quansys because my grandfather actually founded it and asked for some help. My background's in business management, and I had started the restaurant, and I've seen some success there. It's definitely different, but there's a lot of aspects of businesses that are similar. Mm-hmm. Making sure that you provide a good quality experience and product for your customer is key in both of them. And, making sure that you're turning your customers into raving fans, people that want to come back over and over and over again. So in food, if somebody comes and eats and they had a good experience, enjoy the food, they're going to come back, they're going to tell their friends. In multiplexed ELISAs, it's similar. We'll work with customers, and some of our best customers have come from referrals.
Yeah.
So they had a good experience. We focus a lot on customer experience and support at Quansys, and we get some great referrals because of that. So I'd say that part for sure. And then just generally working with people, making sure that I try to treat my employees right so they treat my customers right is something that is the same no matter where you're at.
Yeah. And so do the employees at Quansys get free pancakes?
No, but they should. That's a good. I've brought some stuff in and cooked pancakes for them on occasion, but not too often.
Gotcha. Very cool. Well, thank you so much, Adam, for taking the time to join us here on the podcast. I think it's amazing what you guys are doing, and learned a lot, and glad that there's folks like you and your company out there trying to make the world a better place.
Thanks. I appreciate it. And thanks again for having me.
You bet. Cool.
Is that all right?